and come. So recording, right? Okay, yeah. It probably is a warning for me. I think it's right. Okay, that looks good. Okay, all right. Once again, but now here's a very interesting chapter. And I was telling everybody, if you watch the chapter of the day, we are going to have a video. We may end up showing it that week. If we get ahead, we may do it that week of the 30th or whatever. If I've already done the lecture, I may show it. It's about a 50-minute movie, maybe a little bit more. So I don't know if I can get it in here. But one of the favorite, when I first came here 30 years ago, Pat Miller, who taught this for years, got me to get my job here. She said, you got to take this home, because she knew I was a human biology person. I wasn't really an animal person. I never had a plant class. Excuse me. I didn't know anything about plants. I did my master's in reproductive physiology, and I'd taken all the human stuff. To be a doctor, they never took a botany class. Now you can't get away from it. Y'all get some botany in here, right? So you have to learn a little bit. But anyway, the name of the movie is really got a weird name: Sexual Encounters of the Floral Kind. Yes, I knew some of y'all could appreciate this. So this is the name of the movie we're going to watch: Sexual Encounters of the Floral Kind. Floral with flowers, not any other word that rhymes with that. Story goes, I walked in the first day of class, went over the outline one time and said the wrong word there. Had like 12 people in my class, but that was in an hour. And I had to go back and apologize. That was not the movie we'll be watching. But it's actually going to talk about what we're talking about today, about how the pollinators. I mean, I'm telling y'all, I was impressed. I would have been a botanist if I'd seen this as a freshman, okay? But it's going to show all the different ways the bees and all these other insects and animals are being tricked into being pollinators. Okay, and it's fascinating. I mean, it's a game changer. Okay, and I have it on DVD now, and I think we got it on recording somewhere out there now. But we have it in the library, and I'll get it and we'll show it to y'all. But it's pretty cool. We'll make it a bonus if we come in outside of class. So if we do it like one night, we'll have popcorn and have a little. Because I got a theater down there in that next room anyway, right? So we go down here and watch. It's really good with that surround sound. It's got the 70s soundtrack, kind of like the 70s TV show. Got a really bad old 70s sounds. Anyway, I think it's fun. All right. So anyway, so flower and plants and growth and response is this chapter. This is a really good one here, because we get to cover some stuff that I didn't know existed until I got into graduate school, because I never had I have to take this. And we used to not teach general biology. I took zoology, but now everybody has to take general biology or zoology and botany class. So some schools do a one semester cell biology, animals, and then plants. So everybody does a little bit different. But we're going to look at plant hormones, which is fascinating to me. I think this is a fun chapter today. Y'all will be like, so I got stuff that you can tell your parents about. Okay, I'm telling you, this is some really interesting stuff that I didn't know about. And then plant growth and movement and how they respond. And then we're going to talk about how they have photochromes, which allow them to determine day and night. And so they actually have biorhythms like our. Once again, I have to go back to my master's degree, but my favorite cycle is the female 28-day cycle, right? Okay, I mean, that's a classic one involving hormones and stuff, and a clocking mechanism, right? And so our brain, our gland, the pineal gland and melatonin is where we clock time. The plants have receptors that detect day and night, and that's how they know when the flower is the right time of year. You ever thought about that? How they know? How did the grass know to bloom in the summer and spring? So we'll see some of that today and give you a clue into how that works. Okay. All right. So anyway, right off the bat here, look, your flower plants perceive, and they don't. I don't think they perceive, but they do. I don't. That would imply consciousness. Okay, but they do. I guess they perceive things going on in the environment and they react to them. Okay, so they don't have a brain. Okay, so they don't have a nervous system. But by golly, some of the things we're going to see today makes you think they might have something in both, right? But they don't have a brain and spinal cord, and they don't have nerve tissue. But they can stimulate. They can respond to light, to gravity, to carbon dioxide levels, to pathogens infecting them, to drought. And my favorite one is they respond to touch, right? Y'all all know tomatoes grow better if you put them in a cage, right? They like to climb up a cage, and others stop growing when they get too close to each other. So they will stop, and it will make them stop growing. So they won't destroy the ecosystem, right? And grow too much. So they have contact inhibition a lot of times. They stop growing. So they respond to stimuli that leads to survival of the species. Okay. Responses can be short-term, like the stomata opening when the sun hits in the mornings, when they start doing transpiration. We talked about right before the water up the tree, which is I thought was a pretty good lecture today on that, on how xylem works, right? And long-term, look at this. The response to gravity causes downward growth of the roots and upward growth of the stems. So they have a structure we'll see today that allows them to detect gravity to know, unlike microwave popcorn that says this side up, right? The seeds don't come that way, right? So if you ever plant a garden, it doesn't have a little this side up seed label. Oh, anyway, so you throw them in, but they know they're able to have gravity detecting receptors that tell them which way to shoot the roots and shoots. Okay. Now I'm not going to go crazy with this, but they have to have single transduction, and that's something we talk about in the chapter on hormones in humans or in animals. And what that means is that a little response can cause a whole like a cascade of events to occur. So to do that, you got to have a receptor that takes the sunlight or gravity, a pathway, an enzyme pathway possibly to lead to cellular response. Okay, and I thought that was probably not necessarily needed. Now, wow, how many of y'all do plants have hormones? Okay, I mean, honest to goodness. Okay, I did not know that when I was 19, 20. I did not know that. Okay, I didn't learn that in graduate school. Okay, when I had to go back and I tried 3 PhDs. I didn't finish any of them. First one was in reproductive physiology that I turned into my master's degree, and then I went back and worked in aquatic toxicology and had to take a bunch of. I would actually go over every Thursday and take a class, and then I worked on microbial physiology that I didn't finish, and that's only because I had kids and it didn't pay anything. I think I made $50 a month more if I had that other degree here. So I just kind of let that slide. But what are hormones? Okay, hormones in general in us are chemicals that are made by cells called endocrine glands that get put in our bloodstream and travel somewhere else and make a cell do something that would not normally do. So same concept here. Hormones are chemicals made by a special cell that are transported through the plant's xylem and phloem to another cell and makes it respond as that to that chemical communication. Okay, so they're made in one part of the plant, and we'll actually talk more about human pheromones in the human physiology chapters. And travel within the phloem or from cell to cell and causes an appropriate stimulation. Okay, so here's how one of them works. One of them is called auxin, and it goes through a carrier protein, and it goes through the nuclear pore, binds to receptor in the nucleus, turns on genes, and causes defense response. Response includes bending of the stem. Response includes growing roots and shoots, and growth of the roots, and defense mechanisms triggered. So it turns the gene on or off. Okay, they're pretty wild, and I think they have these. Okay, and then others have receptors, right, that are photosensitive that then cause the chemical to be made inside the cell that turns on or off genes. And here's another receptor that doesn't come in, hormone that binds to a receptor that triggers an internal event. Very similar to the way hormones work in us, actually. They're actually 2 groups of animal hormones. One binds to a receptor on the surface, other goes through the phospholipid biomembrane of the nucleus into the nucleus and turns on the genes. So kind of a little parallel stuff going on here, okay? In A&P, we learn about 25, 28, maybe 30 human hormones, 25 to 30 human hormones. Y'all get a break in plants. We're going to do 5. There are 5 big groups. Now, inside these, there are multiple ones. We're just going to go with the 5 big groups, and this will be really cool for you to know. Okay, and here's our first one. Okay, auxins. Okay, pretty cool. Now, these are produced in the shoots, apical meristem. Now, remember, everything above the ground is the shoots, right? And the apical meristem is the cells that can still divide at the tip of the stem. So this is the top of the tree or the end of the stem. Am I with me? Now, when I start doing this, I'm a tree. Okay, so here's the dirt. And yes, I've got a lot of dirt here. Okay, I've got pretty fat. Okay, and then here's my roots, my limbs, my feet, my roots, and here are my leaves out here. Okay, so we're talking about on the ends of the stems, this apical meristem. Now, the effects of auxin is on growth and development. Okay, it produces the apical area produces auxins that prevent the growth of an axillary bud, which makes them stop flowering at the ends of the limb. So it's going to stop. Apical dominance means it's going to stop growing. Okay, on the ends. But look what it does. It's going to promote the growth of the fruit and the roots, and then prevents the loss of leaves and fruit, and it promotes positive phototropism of the stem. The chemical, so let's say the window is here, and I want to grow toward the light, then it'll make auxins be produced on this side of the plant that will make those cells grow. So it makes the stems grow, just not the ends of the stem, okay? So the plant will grow toward the light by growing the auxins on this side, causing them to reproduce more of the cells. Isn't that cool? Now I have an interesting, fun story about auxins. So when I did that PhD, when I was working on that doctorate degree in aquatic toxicology, I was a PhD student for this guy, and we were doing a project where we were going to go monitor TVA spraying the Garnerville Reservoir in the Tennessee River. So TVA controls the Tennessee River and down through there, and then they actually had Garnerville's big lake like Aqua Marina, and it had somebody had dumped an aquarium back in the 60s, and the hydrilla, which is aquarium grass, and this other called Asian water milfoil, which we think was brought over by barge from another country. They are naturally not native to North Mississippi or the southeastern United States, and they overgrew and took over the riverway to the point where the barges couldn't go through. You could not even ride a jet ski across. Well, you might jet ski, but not a boat, because these things were miles wide and very thick aquatic mats of plants. And so back in the 60s, they started trying to spray it just to keep it open, like where the barges could come through and you could go to the other side of the lake, with your ski boat stuff. Side benefit is this turned Garnersville and other lakes into some of the best bass fishing in the world, because bass love to live in these mats. So our job was to go and to put fish and clams in the spray areas so that when they came in with helicopters or airboats with spray rigs, to make sure they didn't kill what they weren't supposed to kill. Make sure they killed the plants, but didn't kill fish and mussels. And we water sampled 24 water treatment plants all the way to Knoxville. We'd drive and take water samples from their water wells where they were pulling from the river. So it kind of made me feel good that TVA was paying OMIS to make sure they weren't killing people in Alabama or hurting the natural plants and orchards. I mean, the mussels, they don't have their mussels that look like one. Okay, so we get this project. It starts like March 1st, and we got a boat yet, and we got to be over there for a spray in like March 3rd. Now we ordered a boat and we ordered all this stuff, but I had to find a boat to go do this first sample. So I have a friend, a former roommate that lived in Huntsville, so we would go by and get his ski boat. And so I pull into Shoney's like on a Monday morning at 7 o'clock, and all these TVA water boats, airboats are out here, and they're going to spray this code. And we got to go out there and put these samples out and stuff. And so I walk in, I never met the guy running the project. So we sat down at the table, and he ended up being a good friend. The first time I met him, he goes, nice research boat, nice ski boat. And I'm like, well, it's my friends, and our boat's coming in next week. We talk about boats. And he says, today we're going to be using 2,4-D. How many of you have heard of 2,4-D, real famous herbicide? Okay. And he said, we're using 2,4-D. And I said, who's in charge of the project? And I said, I am. I'm working on my PhD. And here's my best friend, Arthur. He's working on a master's degree. So I'm going to get a master's or PhD out of this 3 or 4 year project. And they're like, okay, cool. And he goes, well, who's in charge? So I am. He goes, well, so I got a question for you. Let's see if you know your stuff, because you must be an expert in aquatic toxicology. I'm like, not really. I'm just starting in this field. And he goes, how does 2,4-D work? And I thought, in a big interview like this, first time I meet somebody, do you lie? Do you bluff? Which I have done in the past at times. Sometimes you got to go, Kenny Rogers, you got to know when to hold them, know when to fold them, right? And I look at this guy, and he's like 55 my age now, and I was like 22, and he's like 55. I'm like, I ain't bluffing this dude. He's done played too many cards, okay? And so I say something without incriminating myself. It's one of my best statements ever. I go, we don't know. And he looks at me, he turns red. He grabs a cigarette and goes, damn, you are pretty smart. He said, isn't that funny? Nobody knows how this works. It's the #1 herbicide in the world, and we really don't know how it works. And I'm like, damn, I got lucky. And I go, ain't that something? I have no clue what he's talking about. And come to find out, they did not know how this worked. But he said, but we think it's probably a synthetic oxide, because chemically it looked like oxide. And I go, yeah, I read that. Yeah, of course. But we don't know for sure. So he really thought that was good. It was a trick question. Yeah, so me and him became real good friends. He offered me a job. I didn't take it, I guess. Or should I have? No, I'm enjoying what I end up doing. And I'd rather teach. But anyway, it's a synthetic oxide that look, let me tell you, look, we went out there and put those samples out. They came with that airboat. The next day I couldn't find a freaking plant. Okay. And now I went home thinking about how powerful these hormones are. And I give you one job, I'll say this is the relative of the defoliant used in the Vietnam War when they would spray the jungle, and the next day dude was standing there with a rifle on the top of a tree naked with no leaves, and we'd shoot them out of the tree. Okay. The North Koreans or whatever, the bad guys, the good guys, I don't know who was writing all that, our enemy. Okay. So this thing killed all the plants. It made them blow themselves to death. They blew themselves to death overnight and blew up. And the river was just barely trickling in it, but it blew almost all the fragments away. So I had to work hard the next day and find the root and find plants. They got trapped in the water. So hormones can be quite powerful. Okay. So it does promote the growth of the roots and prevents leaf and fruit from falling, okay, and movement, but it can, but it does cause the stems to grow, okay. Here's what it looks like. So it is very chemical. So 2,4-D was made randomly and found to be very similar to auxins. Okay, here's the other one we're going to mention. Is this some weird? Was your gibberellium? Okay, which I always like to say was discovered on Gilligan's Island, which it wasn't. That's a lie. Okay, cytokines, abscisic acid, and ethylene, which isn't weird. This is a gas. So a lot of y'all think chemistry, right? But here's a gas produced by plants as a hormone. That's pretty cool. All right, and we'll go through them here kind of quickly, and we'll come back to that picture going through. So auxins, very, very interesting. Okay, here is it being put on the root tail on the top, and it stops the growth, right? You can see what they're doing there. They cut the top off and put a little, dip it in some agar with auxins, and it stops, and it won't grow, but the roots will grow. Okay, auxin causes the stems to bend when a stem is exposed to unidirectional light. Auxins move to the shaded side and cause those cells to grow, and it talks how it does that. And then synthetic auxins are used for herbicides to control weeds, okay, which I had experience with that. And they even come in with helicopters and spray them sometimes. We had a lot of fun. I got to ride an airboat and all that. And one time we were driving on the river, and I was doing about 30 miles, 40 mile an hour once we got our boat. And I would pick up the prop and slide over the plants, and they might be, 300 or 400 yards max. And one time I didn't get the prop up in time and threw my partner like 40 feet into the water. I mean, I hit the head of the wall. Okay. He had 3-second warning. I said, hey, I don't think it's going to get up. And by the time we turn around, and I caught myself against the edge of the boat. Agent Orange, that's the word I was looking for, is a synthetic auxin used to defoliate the forest in Vietnam, and 2,4-D is a derivative of that. So very similar to that. And we didn't ever see this in the water table, but obviously it was in there. Okay, and we didn't hurt the fish. We put minnows in cages and kept them alive. And we also had the local residents. We actually found a local mussel diver. And I don't know if y'all know this, but there's probably maybe 15 people over there in Garfield making a living off of going out to the lakes and the rivers and picking the mussels and selling them to the Japanese. And they like to put them in they like to use the American shells of our mussels to make the grit that they put in an oyster to grow pearls. So they would buy this, they would grind it up into little BBs, and that's what they put. They put a BB, little microscopic BB into the gill of anyway, one of the structures inside the shell, what's it called? I'm having a brain freeze here. The mantle. And so a pearl is a sore in an oyster mussel, and they just seal it off in a beautiful sphere that we call pearl. So pearls are made artificially by putting air into their mantle inside their shell, and then harvesting it like 10, 15 years later, maybe less. Isn't that cool? You didn't know. And they like that. And so this guy would. We went out diving with him, and then he wanted me to pay him. He would not take money. We offered him money. He would only take Budweiser. So I had to, I'd take him to the bar, and he would get drunk for 2 or 3 days, and then he'd sober up and go dive for 2 or 3 days. He could make $400, $500, $600 a day diving for these pearls, for these these mussels that look like, native southeastern United States mussels. But all he asked me to do, and I always thought this was really cool, he says, when y'all write the report, tell the truth. Just tell the truth. Do your job, because if it's hurting the muscles, I want to give them to you free so that you're going to protect these muscles, and that's what we did. So we would take their body tissues and never could find the herbicides in them. So it was not going into them. Okay. Later on, when I came to work here, they had succession in that lake, and those plants all died. And then the Bass Association local fishermen sued TVA, and my friend had to testify in court. I had to sign a deposition that what happened. So we proved that they didn't hurt the ecosystem. So they got sued by the fishermen later, years later. Okay. So it's kind of, you never know. So we were doing serious work there. Oh, by the way, you've never lived till you worked in Alabama a whole year in an Ole Miss truck. That did not go well. We got a lot of middle finger, hi, I love yous, and what are y'all doing over here, kind of thing. Because it should have been. I don't know why Alabama or Auburn didn't do it, but I guess we underbid them. All right, gibberellium, another growth promoting hormone, causes stem elongation. There are over 70 different gibberelliums, each different slightly chemically. Most common one is gibberellic acid. It is used commercially to induce growth in crops. And I didn't pull one here, but I read somewhere like something. I'm just gonna make this number up. And in some other books I read, like you can take 2 ounces of this on an acre of sugar cane and increase the yield by like 500%. I mean, this is sprayed in fields to increase the yield. This is going to cause that stem, and that's where the sugar is in a sugar cane, is the stem. Dormancy is a period of time when plants growth is suspended. So this will break dormancy of the buds and cause the seeds to hatch out. It's also sprayed on grapes. So here is the grapes that you grow in your yard or your backyard. I have a friend of mine next door neighbor in Lee County where I'm from, has grapes. His grapes look like this. I've been trying to get into my revealing for years to spray it. So this is almost like steroids for, pump you up, right? Look, come on. So how many of y'all, I got a bag of grapes in the refrigerator house. They look like they're mutated, freaking big as my thumb, right? That's abnormal. That's not normal. They probably spray gibberellin on them to make them real big. So you're eating steroid, and it's not steroid, but I use that as a metaphor. You're using hormonally induced fruit. Okay? We want natural fruit. Go to Whole Foods, right? I'm sure they got the little skanky looking grapes right in there. How many like the big grapes in Walmart? Yeah, me too. I get some feedback from y'all, okay? And look at it over here. Gibberellin causes stem elongation, which decreases the size of the grapes, and it increases the stem over there in growth. So spray that in some sugar cane, and boy, you got something going on there, okay? Cytokines, another class of hormones that promote cell division. This is a big one. It causes the cells to grow. Found in dividing tissues of the roots and in seeds and in the fruit. Have been used to prolong the life of flowering cuttings. So some of these things you buy, the little mixes that have some of that in it, you put in the water for your flowers. Oh my gosh, it's coming up in it. When is the day of days? Valentine's Day. What day is that? 14th. That's coming up next week. Oh my gosh. Okay. So y'all just gotta let me have a moment here. Has there ever been anything stupider than Valentine's Day? Let's be brutally honest. It's already another video, but let's tap it in your class. We cut and sexually mutilate fellow earthlings and give it to each other of the opposite sex. Sometimes same sex, I don't care, I'm judging. As a gift in multiples of 6 or 3 or 12. If I were an alien coming by in my Darth Vader Death Star, I would just blow this planet straight up. Okay, because like that's cray. Why are we sexually mutilating each other and giving them to each other? You understand? That's the ovaries. That is the when you give a girl a dozen roses, you're giving her the cut off genitalia of another living human, another earthling. You understand? Right? Flowers, that's what we're doing. I mean, if you ever give a girl a flower, we give it. Got flowers, come on. It's not disturb y'all a little bit, just a little bit. It doesn't disturb them. Okay. All right. Okay, it's kind of weird. Okay, so anyway, y'all can watch the video. I think we talked about it in the video. But anyway, I think it's pretty crazy. So that's what fruit are. That's what flowers are. Flowers are the fruit. The ovaries are fruit is right in the ovaries. Okay. Okay, and maturing out, die. That die eventually. Come on, man. The flowers fade, right? Don't they? Eventually, unless you do the silk flowers. Okay. Anyway, here is something that plays a big role in that. So it prolongs the flower from having to die. Then die long. Interacts with auxins and cytokines, prevents senescence. Is the aging process of plant. So this is an anti-aging hormone. So this is what the plants need that want to grow. In autumn, low levels of cytokines cause the leaves to change color and to die and fall off. So the fall off foliage that we see the colors are other photopigments that come through after the auxin levels fall. The cytokine levels fall. Sorry. Pretty cool. Now look, I had to look at this 5 times, but this doesn't do a lot for me. This particular picture, I was quite confused. Okay, which I easily am. I'm trying to get the full picture here. Hang on. I don't know why, but I can hyperlink here. Anyway, long story short, let me listen to the hyperlink. Yeah. So I'll go back to the picture because I had to look at this. It's crazy. In this tissue culture that has the unusual amount of these 2 hormones, tobacco cells develop into a callus of undifferentiated tissue by putting auxins and cytokines on it. Okay, so we got a piece of tobacco cells, put it on it, and you get undifferentiated mass when you put both of them. And the point of this little picture is to show you that the hormones interact with each other. Okay, picture B was a ratio of auxins or cytokines that is appropriate. The callus produces roots seen in the downward thin extensions. Let's go back to that. So there's the roots coming down. See them right there? The downward extensions. And that's the undifferentiated mass when you get both hormones. Auxin did this. And then the other one was a ratio, flip the ratio with more cytokines and auxins, and you get the second one. And then the last one is another ratio causing flowers to shoot when you jack the hormones another way. So we can make roots, we can make it flower, or we can make it flower when you're making do it. Here's flowering here, and here you get a lot of vegetation. So these hormones are these 5 groups are used in tandem to get the plants to do what we want them to do, or that what nature, God wants them to do in their development. Once again, you can go back and look at that. I love how they don't put you had to go flip to the back there, sorry. Okay, now we got the last 2 are inhibitory ones, I believe. Abscisic acid is produced by any of the green tissues, contain chloroplasts, sometimes called the plant stress hormone. It initiates and maintains seed and bud dormancy. And what does that mean? That means that the seed is shut down, not trying to hatch out. It's in its state of suspended hibernation. So think about this. The seed is a baby embryo with enough food to last for centuries, in some cases, in a sealed shell. Okay? So when you buy those seeds for your garden this fall, or this spring, next couple of months, my grandparents just put them in the freezer, the one they didn't use. So they'll last several years. You want to save money. That they can they'll. Now, I think I told y'all in one of the lectures that they have found that the Egyptian pyramids, they would kill some of his helpers and his wives to be buried with the pyramids with the pharaoh, so that he'll have his wife in the next life, and then they would have jars of seeds and stuff to grow fruit, and some of those still germinated. So we have got some plant that pharaohs ate has been regrown and sold commercially. Okay, pretty wild if you ask me. Abscission is the dropping of fruits and leaves and flowers from a plant. Okay, and this one is going to trigger that also, I believe, right? And then mutant corn, I don't know what more about the mutant corn stuff. Okay, we'll break dormancy. So this is one that makes the plants prepare to go into dormancy and closes stomata at night. Okay, pretty well. So it just shut down the plant. Here is the leaf change color we see when the cytokine levels are low, and seasonal change, seasonal drop in the cytokines that leads to senescence of the aging of the falling of the leaf. Here's a cool one here. Check this out. Here's a mutant showing the germinated. Look, it's called this corn has a mutation that prevents the hormones that stop the roots from growing to break dormancy, and these seeds germinate on the cob. That's wild, isn't it? So that's roots coming out right there. That's shoots and roots. There's a roots coming out. So that is a mutation where the hormones get knocked out. So dormancy is stopped. Okay. And then our next one is looking at how the stomata opens and closes, and it goes back and shows this. Abscisic acid causes the closure of stomata. The stomata is open to the left. The hormone comes in, and what happens? Abscisic acid binds to a receptor in the guard cell, and that leads to the secondary messenger calcium being made, and that causes sodium channels to open, and sodium is going to exit the guard cell and close the stomata. Remember, a lot of times when sunlight hits the leaf, it actively transports the potassium in, right? And that causes the water to follow it in. So this thing is like an inner tube. You fill it up with water or like an inner tube with air, and you get a ring. Then when you let the air out, it goes back down, and that hole closes. Abscisic acid triggers the closure of the stomata. Pretty cool. All right, and then here's one I always thought blew my mind. Ethylene. Yes, the gas, ethylene. Okay, it's a gas formed from amino acid methionine, and it causes abscission, which is the dropping of the fruit, okay, and the ripening of the fruit. So ethylene stimulates certain enzymes such as salase, which helps cause the leaf, fruit, and flowers to drop. Also causes ripening of the fruit. Okay, now, it's going to blow y'all mind. But right now, over in Yomni is a supercenter, Walmart distribution center. There's a guy at that factory outside of Yomni that carries an ethylene tank on a wheel. So they pick these tomatoes and grapes and strawberries. All of our fruit is picked green. Okay, like they'll pick these tomatoes green. And you'll notice, if you ever been at Kroger's when they put the fruit out and they're in these plastic sealed things, you see sometimes they come out on a roller thing. It's got like, especially your fruit and vegetables, they'll be like a sealed plastic and thing with a little hose sticking off. That's where he hooks the ethylene and sprays it in, and that makes the bananas turn yellow, and the oranges turn from green to orange, and the tomatoes turn from green to red. So we treat nature by it, so we pick them green a lot of times and use ethylene to make them change color or ripen. Y'all know that? It's pretty cool. Now, there's been one of my favorite stories here is a company called Flavor Right back in the 90s was able to go in and genetically engineer one of the first biotech companies in plants, and they knocked out the enzymes that called delayed tomato ripening for like 2 months. So you could pick them red, and they would stay pretty and wouldn't rot for like 2 months. They ended up going broke. They actually got them commercially available. And there was a big test. It was in Time Magazine, Newsweek, where they got 100 food critics in New York City, brought them in, and they had the flavor out tomatoes, and they had tomatoes shipped from California green that was shot with ethylene, right? And how many, okay, long story short, how many of y'all had those tomatoes that taste like paper? You ever notice that? They don't have flavor. Because to get the flavor we like in tomato, it needs to ripen on the vine in Mama's backyard. Okay, so I'm saying that try that when you get tomatoes in the spring. Tell me, our winter tomatoes always to me taste like paper. But when I get them that I grew or Daddy grew, they have more flavor because they were let to ripen on the vine. Okay, so they did a blind taste test with 100 food chefs in New York City. 90% chose the flavor out tomato, wanted to buy it. But when they told them, this is back in the 80s, when they told them this, they said it's bioengineered, it has a tobacco gene in it, and they refused to buy it. Company went broke, Monsanto bought them out, Monsanto now has that technology. They also made celery that was snappier, and had more snap in it, like altering the DNA of the structures of the parenchyma and spoenchyma, in the ground tissue. Yeah. So Monsanto is probably the largest agricultural biotech company that owns Roundup, or used to own it, and they do all the seeds and stuff. But pretty interesting. So we use that. I hope that guy don't smoke, right? He can blow himself up. I always make a joke about that. Hopefully he's not smoking. But anyway, ripening of the fruit and suppression of stem and root elongation. Look at this picture here. Here is a piece of holly, okay, with no acetic acid, and it will stay like that it will stay pretty. Drop a tomato in there with it, and the tomato produces ethylene as it changes color. And look, within within a week, the leaves have fallen off. So same leaf, put in that water at the same time with nutrient water, but that ethylene being produced here causes the obsession of the dropping of the leaves and fruit. You can buy bags in Walmart. You ever see those little bags you buy that says fruit savers? Keep the fruit, especially like how many of y'all, come on, man, I know it's been a while since Mama packed your lunch. But when you're like in the 3rd grade, Mother would cut up a apple and put it in a Ziploc bag. And you get to school, it's brown and looks kind of gushy and soft, right? Well, this has ethylene grabbers in the plastic, and it sucks up ethylene so it'll be crispy when you eat it at lunch 4 hours later. So that is all about if you go look at the little bags, they're made to catch the ethylene, so the fruit will stay pretty longer. So we got a lot of little tricks going on with that. But I always thought that was pretty wild. Ethylene being a hormone, pretty cool. Here's showing some other ones here about ripening of the fruit and the genes being knocked out. So this talks about how they were able to knock out this enzyme so that the tomatoes, see how the tomatoes ripe and they get all squishy. And now with no ethylene, you can pick them green and then shoot them with ethylene at the distribution center. A lot of these are grown in Mexico too, but most of our fruits and vegetables for winter are grown in the beautiful San Bernardino Valley, east of the ocean, before you get the mountain range. Anybody been to California and seen that? Beautiful. I was at a wedding, and one of my good friends there in Modesto, which is this fruit stand over. We had the reception at a almond farm. Friend there at an almond farm. Okay. But anyway, beautiful fruits. You get fruit stands everywhere, and you get fruit stuff really cheap. I have not been, and this is one of the things I need to do by my wife, and both my daughters have been on the Panama, the country, not Panama City, Florida. Okay, on a mission trip, and my wife probably done 15 times, and they say the fruit down there are unbelievable. They say like you get the bananas and the pineapples that have been ripened on the vine. It's that same wild experience as you would between tomatoes grown, hand grown, ripened red, and the commercial ones we buy. So every time they tell me our pineapples are terrible compared to the local pineapples you get down there. And so a lot of times they'll just do fruit for lunch down there. Okay, lots of fiber. Okay, one of our jokes here in the middle. Gotta have fiber in your diet, right? Anyway, that's kind of cool. All right, now we're going to talk. The next part here is talking about tropism. That's movement. Anytime a plant or anything moves towards something. Okay, the most common one you might think of is phototropism. How you can put a light up and the bugs go to the light, right? Anybody got one of those zapper lights? It's got the electric wire on it, right? I love that. Zap the bugs, kill the bugs in the back porch, right? So plants do phototropism, move in response to light. Thigmotropism is moving in response to touch. And this is kind of cool how that's the tomatoes like to go up the cave, the vines or whatever. And sometimes they stop moving when they touch each other. And gravitropism is in response to gravity. Here's a classic picture of phototropism with the stem bending so that the leaves will be pointed towards the window. In the video I was telling you about section accounts of coral kind, there's a flower found in Antarctica, or it's either Arctic or Antarctica. It's this beautiful yellow buttercup, and when the sun comes up, you're out there up north and up there, and the sun doesn't go down sometimes, about for 2 or 3 months, because of the orbit. And so this thing just sits like this, and this little thing, the GPS tracking, the flower moves and follows the sun, and it reflects all of the light onto its petal on the middle part of the stigma, and it's about 10 degrees warmer inside this flower. So the plants, the insects get in there for a sauna, and so it tracks the sun all day. Pretty amazing. By moving this, by producing hormones in the stem, it's able to track the sun. That's pretty freaking cool. Okay, and it talks about how the photoreceptors respond to blue wavelengths of light and trigger the production of the hormones. Okay, stigmatropism. Here are some beans or tomatoes growing up. This are beans growing up the pole. And Morning Glory also likes to do this all over our fence posts. Gravitropism, when a plant is placed on its side, the stem goes up, and because of the pull of gravity, and the roots go down. Okay, and this is due to sensors called these stoliths found in the organelles called an amyloplastid. I did not know that one. And these stoliths settle to the bottom of a cell, put pressure on the organelles to signal to the plant that that's downward. And here's a picture of that. Really cool picture right here. See the little crystals right here? So this plant is setting up right, okay? And the crystals fall to the bottom, so roots down, shoots up. Now look at this cool experiment. Take the same plant and put him in this little thing that rose ever so slowly. So the crystals are kept in the middle and they don't know what to do. So the plant just goes straight out. Because normally it would grow what? If you knock a flower over, it'll what? It'll grow up, right? The roots go down. But in this little turning experiment here, they keep going straight because they don't know what to do. Okay? Because the little crystals stay in the middle. Okay, little crystals are called. I thought that's pretty cool stuff. That's a new one for me too. I was reading on this. So these stoliths and the little amyloplast, whatever structure that we now identify with there. You learned something new today, right? I learned something new last week reading this one. This is new to me. Okay. Others here, what we're talking about here, nature, nothing much here. This is a really cool little plant that we do a lot of scientific experiments with. Look at that. This is one that's real famous, being used in science to do experiments with, because it grows in 5 to 6 weeks to get an entire maturity. And normally self-pollinates, but it can easily be cross-pollinated, so you can easily remove the male and female parts. And it has a small number of base pairs of DNA, makes it a good one to do genetic experiments with. Okay, other ones here, other stimuli here, tongue movements here, to touch, okay, and talks about that. That's a great one here. I mentioned this in the video for yesterday's chapter, but I actually bought a Venus flytrap one year. So we have a little bit of budget, and so I actually bought a Venus flytrap one year and kept it in the lectorium in that big room over there. In the old building, we had a room like that. I don't know if y'all been in this building, but the PS over there, chemistry and physics lab, there's a big classroom that holds about 100 people. We do speakers and stuff with, and orientation and stuff. And we have one in the old building I worked in. So we built that just to kind of remember the old building. We don't want to be a community college should have small classes like this. But we do want to have one big room, so if I need to have it, we're not going to turn you down in chemistry, right? I'll move to the big room. Anyway, I brought a Venus flytrap in the other building, and we watched it grow, and it said it's a little trap. Now, it doesn't consciously do this. It has a trigger, and an insect lands, and it closes. But this one is like a hair trigger. And we were sitting in class one day, and look, we heard a snap. Everybody looked, and he caught a bug and eaten. And they actually dissolve it and use it for minerals. So they can grow in places like the classroom that don't have any minerals laying around. Okay, anyway, so it's a mechanism. That's a good example of this movement, right? Does it consciously do that? Okay. But we also have the next one, photosensitive pigments called photochromes that can allow them to have circadian rhythm. Now, this is the rhythm that we talk about of like a 24-hour cycle. You know, we get sleepy at night because the sun goes down and we make a hormone melatonin. Well, they have receptors that will detect day and night. And that's very important because the stomatas need to open in the day, but close at night. But in the desert, the stomata stays closed in the summer, in the daylight, and opens at night because it would evaporate itself to death in the heat. So these circadian rhythms are very important. Okay. And it talks about that. So when water goes in, the cells swell up and get rigid. Here, when water leaves, they get thin, and you can see, look at these leaves. They're responding to touch here. The target leaf of the sensitive plant, before and after it is touched. If you touch it, it closes it down, and the leaves will close down. Look at that. So that's before and after by cutting the water flow off the leaves. Why it's doing that, I don't know, but that's what it does. Okay. The photoperiodism then is, here's the photochrome I was just mentioning, that allows them to have this day and night influence on flowers. And this is how they're going to know when to flower. So they have a mechanism of counting so many days, or at least knowing when the day gets shorter, than they flower on that. Okay. Here's how it works. The photochrome is a blue-green leaf pigment that often exists in two forms. The PR, or photochrome R, is the inactive form, so it's not going to trigger the cell to do anything. The photochrome FR, far red, is the active form. So when certain wavelengths of light hit it, it becomes active or inactive. Okay. And the variation in those two forms are detected and promotes the cell. For example, seed germination and flowering, and it can inhibit the shoot elongation. So this actually will cause seeds to germinate out. The germination is not meaning to become a German citizen. Seed germination is when you get water and moisture in and oxygen, and you hatch out the baby and grow the baby flower out. So you put the seeds in the ground, germination occurs, when you water it, it rains, and then it comes out. Okay. And this can help trigger some of this. And here's how it works. Take a look. The inactive form is converted into the active form in the presence of red light. The PFR active form is involved in various plant responses such as germination, elongation, and flowering. Okay, and it is converted back to PR whenever light is limited or in the shade or at night. Okay, this brings us to these very cool short answer questions on your list. There are three groups of flowers based on their flowering status. Okay, let's check it out. There are those that are called short day plants that flower when the day length is shorter than a critical length of night, or a critical length. So when do the days get shorter? I remember we talked about this maybe, I think we may have talked about last semester. But one of the cool things about nature is that our Earth is orbiting the sun, and we have the movement of our planet in its axis. So the longest day of the year is in like June 15th, something like that. The shortest day of the year is usually Christmas Eve or right around Christmas. So days are getting shorter in the fall, right? So right now the days are getting longer. Everybody knows the days get longer. Days are getting longer right now, and they're gonna get longer to like June. Okay. So flowers that are waiting for a day length to get longer, flowers when the day length is shorter, these are gonna flower in the fall. Now, how many of y'all notice that right there are two big times I have allergy meltdown? Spring and fall. So the fall plants are the ones that flower in the fall, are short day length. And the long day length flowers when the day length is longer than a critical length, and then they flower. And that's the ones that are going to hit us here about March, April. Of course, with global warming, it's almost year round now pollination, right? But we have seen warming going on. So there are those that flower in the fall, the short day, and the spring ones are long day plants. And then day neutral plants do not depend on this, and you just plant them and they flower accordingly, right? Like corn, you get 120-day corn or whatever. Okay, so some of them don't have that, they fall in that group. Now watch how this goes. This is really cool. I think I mentioned this in the lecture of Monday. Cocklebirds, okay, are a short day, long night plant. So they are waiting for the day, they're waiting for that short day, okay, and then they will flower. Okay, so when the day is shorter than a critical length, this type of flowering age, the plant does not flower. When the day is longer than the critical length of time, or if it's interrupted by a flash of light, it will not flower. So in other words, this is looking for a long day. It's short. When the day is shorter, this is night long. So the night's got to be that long in that line. So it's not going to flower here. But if the day it reaches that, you shine a flashlight for just a few seconds, you can delay the cucumber from flowering indefinitely for a week or two. I actually seen that experiment done. Matter of fact, you may have done this before. How many of y'all have those cucumbers in your backyard? But I've never seen them come up hardly because you got lights around the house. So that may be how they don't flower. Is that not pretty cool? That's pretty cool. Okay, and then the other one down here, when the day is longer than a critical length, so here the yellow is the day, so waiting on that day to get here, we flower. No, but then you can shine a light on them and they'll flower in the middle of the night. And they'll flower the next day. That's pretty cool. Oh, went almost over time. Okay, here we go. So here's normal photochrome activities, and shows how to elongate. Nothing much here. They can use that for plant spacing. 24-hour cycle. I think we're good with that. Nothing much here. If they just do have circadian rhythm, look, morning and night, they close. Morning glory opens up. By the afternoon, it closes. They use these photochromes to do all kinds of stuff. All right, y'all have a good day. Appreciate y'all. Hope that was pretty interesting, right? Just a little bit. Okay, thank y'all. Couldn't tell. I thought y'all were getting mad at me for a minute, like it's boring. I'm sorry. Fun job is boring a little bit. This is, to me, is a little more interesting.